final presentation in this session will be given by Brian Yaspan from Genentech, Roche, and he will talk about whole genome sequencing efforts of IPF clinical trial patients. Brian. Stage is yours.
My name is Brian Yaspan. I'd like to say thank you to the organizers for letting me be part of this really good session. These have been really good talks. So I'm going to talk to you about some of the work that we've been doing on our IPF patients' DNA. So kind of wanted to start with what are we doing at Genentech with genetics. It might not be so intuitive to have a human genetics department at a biotech company. So we're using information from the human genome to discover and verify biomarkers, to understand disease pathogenesis and heterogeneity, and to discover drug targets. And we do these in various ways, but these are kind of broken up into three main screens that we're doing. So first we have what we're calling like risk and protective alleles, comparing cases to controls, looking for factors that predispose to disease. We can also look at what we kind of call modifier screens, and I'll get into this later in the talk, but looking at patients within the disease of differing genetic burdens, so high versus low genetic burden, to homogenize the patients and see what makes them different. And finally, obviously, pharmacogenetics, where we can subdivide patients based on risk SNPs or other types of SNPs and look for patients that respond to disease, such as on the top, you would have or do not respond to disease. So as has been covered earlier, but so I'll go kind of quickly through this. IPF does have a substantial genetic component. Familial aggregation studies have seen variation in surfactant proteins and telomerase. However, in terms of the whole, these individual variants account for a small portion of the overall population genetic risk for the disease. But they do, and this will be important for the whole genome sequencing, they do demonstrate that infrequent high penetrant variants have a strong effect on susceptibility. So here, after the familial studies, the genetics field went into what we now call the GWAS era. These are looking at common variation in individuals. For those not familiar, this is called a Manhattan plot. This is the results from a large IPF study published a few years ago. You array let's see, there we go. You array the SNPs by location on the bottom of the genome, and then p-value on the top, and it's called the Manhattan plot because you're looking for peaks, tall buildings, let's say, that pass this statistical threshold of genome-wide significance. So here we have the risk alleles that have been seen. As you can see, the MUC5B SNP rises to the top, and then you have other SNPs that, while the MUC5B SNP does have a large effect size, these other ones have odds ratios in lines with what we would expect from common variant genetic studies. So odds ratios are around 1.3. And this has also been shown earlier, but oops, that was supposed to come up as a reveal, but I revealed it prematurely. You have your allele frequency on the bottom, increasing allele frequency, and effect size going up. So the rare variants identified in familial studies have a much larger effect size, but the common variants affect a larger swath of the population. So they do have an effect down here. The MUC5B SNP is unique in the regards that it does affect risk in a substantial way. However, it is common in the population. So what are we doing at Genentech in order to look at our patients? So we have what we're calling the HGI, the Human Genetics Initiative, where we have sequenced about 27,000 patients so far from our clinical trials and from collaborations. And I highlighted the IPF samples here, so we have a little over 2,200 IPF whole genome sequences. This is a very large amount of data, about almost 9 petabytes and 81 billion genotypes. So these numbers were kind of abstract to me, so I asked our IT department, what exactly does this mean? And so they came back with a single BAM file, which is what you get off of the whole genome sequencer. Is about 56 times the size of a 90-minute high-definition Netflix movie. So this also didn't make much sense to me because I have a toddler and I haven't watched one movie, much less 56. But I actually realized that I have. It's just not a movie that I wanted to see. So for Thomas and the Magic Railroad, how does this relate to DNA sequence? 56 showings of Thomas and the Magic Railroad is equal to one person's DNA. And our current data amount is a little over 1.5 million showings of Thomas the Tank Engine. When I showed this slide to my son, he said, "That's a lot of Thomas." So enough of that. So how does this different from the GWAS chip the GWAS data that we've done? So as Jonathan said, GWAS chip data looks at common variants. It's a kind of a canned assay. It's on a chip. You put the chip in, put the DNA on, and you get your results. This is made possible because of the structure of the human genome, such as that SNPs closer together tend to be inherited together. So you don't have to tag all of the SNPs, or genotype all the SNPs in the genome, but you can genotype select few SNPs. And so these chips have about 2.5 to 5 million SNPs on them. There's many pros. They're cheap, reliable, reproducible. You get very good coverage of common variants, but the con is that you do not get these rare variants that we've been talking about in this session. And you kind of arbitrarily define that as less than 1% frequent in the population. So this is actually a big deal because this rare variation adds up in the human population, such that about 2/3 of the total variation in humans is this rare variation. And it is more likely to include the protein-altering variations, missense, nonsense mutations that will directly affect a protein. Whole genome sequence data, on the other hand, is able to call that because you have millions of short reads. You align them to a reference genome here, and you call SNPs on the fly. It's not a canned assay. You just go across the genome, and any place that's different, you can call the variant. So you end up getting this rare variant coverage. There are cons, and this is obviously pricier. As I've mentioned, it's a lot of data, and it is very sensitive to batch effects. So once you get the whole genome sequence data, how do you analyze it? Well, these Manhattan plots, every dot is an individual SNP. And so you can do that because in your cases, everyone has the same SNP, and your controls, everyone has the same SNP, and you compare them SNP by SNP across the genome. For rare variants, it's a little different. Patients might have, and if this is a gene here, they might have a variant here, 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 but nobody's going to have, not enough people are going to have the same SNP to compare them SNP by SNP. So what you can do is take the gene area and collapse it and do a burden test such that how many cases just have the presence of a variant compared to how many controls. This actually, when you add this up, it does, you are able to do good analyses on this. This is an example from macular degeneration. There was one locus called complement factor I that had an odds ratio of what we expect from the GWAS studies, as I mentioned, of 1.14. So people weren't all that, it affected disease risk, but not substantially. But when we started looking at rare variation, and this is the published data here, 8% of macular degeneration patients had a variant in CFI compared to only 2% of controls. And then we replicated that in our Genentech AMD trials. So this elevated the status of that gene and has opened up new lines of thought regarding this gene in pathology of the disease. So we're seeking to do this in IPF. These are the numbers of the samples of how many sequences that have gone into the data that I'll be presenting from our pirfenidone trials, the INSPIRE interferon gamma, lebrikizumab trials, and observational cohorts at Vanderbilt and UCSF. And so I have some examples of these different types of studies that we've done. So for disease risk, again, this is looking at the burden of these genes. So we started with a candidate gene analysis, looking at our IPF patients and our non-IPF patients. And we selected candidate genes that were seen in familial and the GWAS studies. And what we saw, as had been mentioned before, the genes RTEL1, TERC, and TERT had an enrichment of rare variation in our IPF patients compared to non-IPF patients. So TERT was especially interesting, obviously, because it has a common variant and it does have rare variants associated through familial studies. So we looked a little bit more into this, and we decided to start by increasing the stringency of our allele frequency cutoff. So from 1%, which is rare, going to ultra rare, and then ultra, ultra rare, if that's a word. So increasing the stringency also enriches the signal. So what we're starting with is a minor allele frequency of 1%. We see an effect size of 2.8, with 4% of IPF patients having a variant compared to 1.7% of controls. When we have that, go down to 0.5%, we attenuated a little bit in IPF patients down to 3%, but pretty much remove it in the control population, down to 0.2%. And this increases the enrichment to 28, and then further, if we go down to 0.1%, enriches it to 44%, odds ratio of 44. We also did this for TERC and RTEL1, but these variants were already so rare that this didn't change the stats at all. So we noticed that of all the previously identified risk loci, it was only the telomerase-related genes that showed rare variant enrichment in our population. So then we expanded this to include other genes in the telomerase pathway. The one gene that is missing here is PARN, and we're still working on that. There were some quality control issues with that gene. But we didn't really expand anything beyond RTEL1, TERC, and TERT. So in summary, the rare variation in these three genes is enriched in IPF patients relative to controls. So now talking about modifier screens, and as I said, I'm going to go into a little bit about what I mean by this. So in terms of genetics, IPF does kind of gravitate towards this kind of study because you have this very strong risk SNP at the MUC5B and TOLIP locus. In the general population, about 19% of people walking around carry this variant. However, not very many of them end up getting IPF. So why does this happen? And furthermore, when you look in the IPF population, 60% of patients have a variant here, have the risk variant here, but 40% do not. It's just absent. Why are these patients getting IPF? So this, furthermore, as has been said, that this is functional. These patients with this SNP do progress differently, such that patients with the risk SNP have longer survival than patients without the risk SNP. So why don't these people get IPF? And furthermore, why do these people get IPF, and why do they progress faster? So we started by looking within our IPF population, comparing patients that I'll be calling MUC5B positive. MUC5B positive patients carry a risk allele there. MUC5B negative are patients that are homozygous for the protective allele. So we did this for TERT, TERC, and RTEL1, and basically only found TERT was very interesting, such that patients with a risk allele in TERT, about 3% of them, compared to about 7% in the patients that did not have a risk allele at this locus. We extrapolated this by genotype, and coming over here, patients that were homozygous for the risk allele had a frequency of telomerase rare variants that was similar to a general population, so 1.5%. And this increased as you took away the risk allele. So as you added the protective alleles, these patients seem to have much more telomerase mutations. So in summary, this variation in TERT is enriched in patients without a MUC5B risk allele. So as also has been said, and we wanted to see this in our own population, these are the different disease patients that we have in our cohort, and this is their telomere lengths here. So we calculated telomere length from the whole genome sequence and found, again, our IPF patients had substantially shorter telomeres at all age points. So how does this relate to the genetics that we found? So if you add up all of the TERT, TERC, and RTEL1 variants, you get a frequency very similar to what was shown in the first talk of this. About 12.5% of patients have one of these variants, compared to definitely about 2.5% of all our others. This was a smaller trial, and we didn't see the enrichment there, but for our larger trials, we are seeing a substantial enrichment overall for these three genes. And again, do our patients have shorter telomeres? And the answer is yes. So this is in all of our 30,000 samples. Patients with no variants in these three genes, patients with one, patients with two. And what we're finding is it's about 250 base pairs shorter per telomere variant. And you can overlay this on top of the common telomerase variant. So that's what we have here. This is the common variant, the C allele being the protective allele, A being the risk allele. And slightly shorter telomeres as you progress to the risk allele here. But when you overlay rare variant status on top of that, you can see that even though all these patients are rare variant carriers, patients with the protective allele have longer telomeres than the others. And then finally, I wanted to talk a little bit about the first analyses we've done for pharmacogenetics. And so we've looked in our pirfenidone trials, so the ASCEND and capacity trials. And here we have divided patients at the median of telomere length such that long has solid line, short as a dotted line, and then black placebo and red being pirfenidone. So we do see, and we see again that shorter telomeres predict more rapid FVC decline in patients. However, pirfenidone treated patients demonstrate treatment benefit regardless of this telomere length status. And similarly for MUC5B is the same type of plot as over here, dividing by presence or absence of the risk SNP. the MUC5B negative patients predict more rapid FVC decline. And this decline was also attenuated in patients regardless of the genotype. So in summary, as we've all said in this session, IPF does exhibit a strong genetic component. An excess of rare coding variants is seen in genes encoding these telomerase components. Telomere length and MUC5B risk allele status are associated with FVC decline. And our future plans here are we're going to be expanding this genome-wide. We do have whole genome sequence data, looking hypothesis independent across the genome for both rare and common variants associated with risk, disease progression, and treatment response. And as well as further trying to homogenize the population and seeing the difference between MUC5B positive and negative patients. With that, I'd like to say thank you to everyone within Genentech and out that has helped with this study. It's a lot of people. And most importantly, thank the patients who generally donated their DNA for research. Thank you.